Another very important organelle in the cell is the Golgi Apparatus or Golgi Complex. Have a look at the model here, and it shows you the structural appearance of the Golgi Apparatus. They're really parallel stacks of tubules and also little vesicles. They are closely associated with the nucleus and also the endoplasmic reticulum, and they have what we call a cis face or an entry face, and a trans face or an exit face. And the reason for that is that one of the jobs the Golgi apparatus does is that it accepts proteins and various components synthesized by the endoplasmic reticulum. Proteins synthesized by the endoplasmic reticulum are packaged into a transport vesicle, these small green-coloured vesicles here. And that transport vesicle then enters the cis face of the Golgi complex, and the components or the products pass through the Golgi complex where they're sorted, modified, and then repackaged into a secretory vesicle shown here as a blue-coloured vesicle, which then passes to the cell membrane. And the secretory vesicle itself fuses with the cell membrane and releases the secretory product. That process is called exocytosis. And besides sorting and packaging, the Golgi apparatus is involved in making carbohydrates. Now you can even see Golgi apparatus in a light microscope image. If you look at this cell in bone marrow, you can see a very pale area near the nucleus. That represents the Golgi apparatus. Here's another example here, and also here. When you look at this electron micrograph, the Golgi apparatus is visible as stacks of membranes. Above the stacks of membranes, you can just make out some profiles of mitochondria, and the darker stained circular structures are going to be lysosomes, which I'm going to speak about in a moment. These small dark dots are ribosomes, and some attach here to the endoplasmic reticulum. I'm not going to mention too much about peroxisomes, except to say that they're involved in the oxidation of fatty acids and hydrogen peroxide. Lysosomes, as you see in the model, are single membrane-bound organelles. Now there's a little bit of controversy as to their origins, and I'm going to describe what has been the traditional function and structure of the lysosome, and then modify that story a little bit. Lysosomes consist of enzymes that are involved in digesting cell components or large molecules. Traditionally, they were described as being bound by a membrane and produced in the Golgi complex, depicted in this model as these orangey-coloured vesicles. And that membrane was to protect the exterior part of the lysosomes and the rest of the cell from the very strong hydrolytic destructive enzymes. These lysosomes often come across or target either damaged organelles or aged organelles. Cells are forever getting rid of surplus organelles, so lysosomes break them down. Lysosomes also break down large molecules that the cell ingests. This could be occurring here. The cell may have ingested some material by enclosing it in the cell membrane. Then a vesicle containing that material enters the cell as an endosome, and a lysosome fuses with the endosome here and breaks down the material ingested. Another function of lysosomes is to break down the cell itself during necrosis. When the lysosome, you see in this model, leaves the Golgi complex full of its enzymes, it used to be called, or at least in some of the older traditional histology teaching, it used to be called a primary lysosome. When it fused with a structure that it was going to degrade, such as this vesicle, it was called a secondary lysosome. And then finally, when it had completely broken down all the products that it's targeted and is just lying as residual components in the cell, maybe this vesicle here, either to stay in the cell or to be gotten rid of by the cell, then they're termed tertiary lysosomes. That was the traditional understanding of what lysosomes are and what they do. Now there's a little change in what the thinking is, and I just want to describe that briefly. The theory currently is that we tend to accept that lysosomes are produced by the Golgi, but they're not necessarily packaged or activated in the way previously thought. They're probably made by the Golgi apparatus as inactivated hydrolytic enzymes, and they target ingested endosomes that carry either material that the cell has ingested, or they will target old organelles, etc., just like I described. So rather than being membrane bound, they're lying loose in the cytoplasm. Endocytosis is the name of the process when the cell membrane clusters around products on the cell surface and ingests some of that to form an early endosome. Now, an early endosome is a vesicle within the cell that has taken in those external molecules from outside the cell. It starts to recycle some of the cell membrane components back to the cell surface, but then, as it's finished doing that, it proceeds to go deeper into the cell. When it's targeted by the lysosome enzymes and they're activated, other membrane components are made by the Golgi complex and modify the endosome membrane so that these hydrolytic enzymes cannot destroy the cell itself. It's then termed a late endosome, or more or less like the old terminology of a secondary lysosome. So really, the only real difference is whether or not the lysosome itself is a vesicle membrane-bound component of these enzymes, or whether the enzymes are now thought to be just free in the cytoplasm in an inactivated form. But then, when they're targeting ingested endosomes, they become activated, and the membrane of the endosome becomes altered so that the rest of the cell isn't destroyed by the activated hydrolytic enzymes inside.